So Photosynq is a platform where anyone can quickly and easily create, share, and analyze plant health information. Our goal is to create a community of people all around the world who are not only collecting data about plants, but also creating ideas and asking questions about plants that no one could currently ask because that network of people to answer them doesn't exist. First, we have a multi-spec, is a device which measures a variety of plant health indicators like photosynthesis, photosynthetic activity, leaf greenness, and a bunch of other stuff. So you would go out of the field and you would press a button and take data with this device, and that the data would then go to your cell phone and then to an online database, and then from that database you could access it from a website. So the website has a bunch of really neat online analysis tools. So now you can compare different parts of the project, you can see your data, you can see other people's data, and you can collaboratively discuss that data from that point. So that's the cycle of data out. The flow of ideas goes the opposite way. You start on the website, you create a project. Within the context of that project, you define the measurements that will be taken. And then, someone who has their cell phone sees your project, right? And they say, oh yeah, I want to find the most photosynthetically efficient algae, for example. So they take part. They follow your directions. They take a sample according to your protocols, and then the data goes back to you. So the idea is that ideas go out and data comes back. And then in the middle of that sits the most important thing, which is the community, right? All the people involved in taking the measurements, coming up with the questions. But because the whole project is open source, everything from the software to the hardware to the data, they're also able to interact at every single level, not just data and ideas. They're able to create new sensors, for example. So maybe right now we can't measure soil biological activity, but someone wants to create that method, they can do that. The device itself costs between $50 and $125 to make. The rest of the tools are all free. So the future implications of this project depend on the field of interest. There's a new set of techniques and methods that breeders are using to make their selection process more efficient, and it's called marker-assisted selection. But they require a lot of equipment to take those measurements. So we're capable of doing that at a very low cost. And this applies particularly to the developing world because they have even less money than breeders here. So I mean, this is a problem for breeders in the United States, but it's certainly a huge problem for breeders in the developing world.